Today we're going to be talking about nondisjunction during meiosis. And nondisjunction is a failure of the chromosomes to separate properly. Now, in order to understand this, we have to be comfortable with meiosis, normal meiosis. So if you need to, go back and review the meiosis videotape. But let's go ahead and get started with the diagram of normal meiosis. So this is how it's going to begin. I'm going to use two different colors here. I'm going to use blue and pink because in a normal cell you have 46 chromosomes or two sets of 23 chromosomes. One set came from mom and one set came from dad way back when you were conceived. And all the cells in your body are genetic duplicates of that one cell. So in a sense, all 100 trillion cells in your body have two sets of chromosomes for the most part. One set came from mom and the other set came from dad. And that's how I'm going to draw it on the board. So let's go ahead and get started. This is going to be normal disjunction. This is how things should proceed if everything happens correctly. So I'm going to go ahead and begin by drawing a tetrad. So there's a single tetrad, and here's the cell right here. There's the cell. Now obviously the chromosomes are not that big in a cell, but we need to be able to see what's going on there. So here's a chromosome from dad, here's a chromosome from mom. And I'm only showing one tetrad, but there's 22 other tetrads in there. So there should be 23 tetrads in this cell. And I'm just going to show one out of the 23 tetrads to make things easy. Now let's go ahead and get started. This is prophase 1 of meiosis. Now eventually this cell is going to go through a type of nuclear division called meiosis. Meiosis 1 is going to proceed. And when meiosis 1 proceeds, our tetrad is going to separate into two dyads. So there we go. And I'm going to draw the little centromere here. And there we go. There's our centromere, and now we have two cells. That was normal separation right there. Normal disjunction occurred. And now these two cells are going to go through a second meiotic division to produce 4 cells. So this is going to be meiosis 2. And now our dyads should separate. So there they are. There we go. Draw our cell around there. And what we end up with is 4 cells that are going to be genetically different from each other. And each one of these cells should contain 23 chromosomes. And again, I'm only showing one out of the 23, but the other 22 are in there. Now, because each cell contains one set of chromosomes, one set of chromosomes is denoted by the letter N. N represents one set or 23 chromosomes. So this has N, or the haploid number. And we could say that it contains 23 chromosomes. So there we go. That is normal disjunction. Now we're going to compare this with abnormal meiosis, or nondisjunction, when the chromosomes do not separate properly. So let's go ahead, and I'm going to erase some of this information. And I'm going to erase what's in here. We'll keep our cells there. I'm just going to erase some of this information in here. And again, we're going to start with a tetrad. And this is going to be nondisjunction during meiosis 1. So this is going to be a failure to separate during meiosis 1. So here's our tetrad. And again, remember, that's only one out of the 23. And we're going to show, because non-disjunction is a rare event, we're going to assume that the other 22 that I'm not showing here are separating properly. So I'm only going to show you the problem with this one tetrad. So here we go. There's going to be a failure to separate during meiosis 1. So what's going to happen is this. Both of these are going to end up moving toward one cell. And the other cell does not get one of these dyads. Now remember, this cell right here, which I'm showing with nothing in it, really has 22 in it now, because we said the other 22 are proceeding normally. So even though I didn't draw anything here, there's 22 dyads in there. Now we're going to proceed with normal meiosis 2. So these are going to separate. And again, here there's nothing to separate. And let's see what we end up with. Here we have a normal set of chromosomes plus one more chromosome. So we get n plus 1. And again, a normal set of chromosomes plus one additional chromosome, n plus 1. And here we're going to have a normal set of chromosomes minus 1. We're actually missing a single chromosome there. And again, n minus 1. What does that mean? We have 23 chromosomes here plus one additional one. So we actually have 24 chromosomes here. Again, same thing, 23 plus 1 for a total of 24. And here we've got 23 minus 1 for a total of 22. And again, 23 minus 1 for a total of 22. So in this case, because the error occurred early on in meiosis 1, all 4 gametes, meaning sperm or egg, that were produced are going to be defective. None of them are going to have a normal number of chromosomes, which is 23. This one has an extra one, this one's missing one, this one is missing one. So 4 defective gametes here. So if any of these gametes are involved in fertilization, we're going to have a child who ends up with, unfortunately, all defective cells, because every cell after that initial cell is going to be a copy of it, and they're all going to be defective. And that's going to cause big problems. Now, if a gamete with an extra chromosome is involved in fertilization, that's going to result in what we call trisomy. So the child is going to have 2 normal sets of chromosomes plus an additional 1 chromosome. That's why we call it trisomy. Tri means 3. There's an extra chromosome. So instead of that child having 46 chromosomes, that child's going to have 47 chromosomes. And a good example of that is Down syndrome, where you have an extra 21st chromosome. Now, exactly the opposite is going to occur here, kind of. We're going to be missing a chromosome. So instead of having our normal 23, we have 22 chromosomes. If that gamete is involved in fertilization, the baby, the child's cells, are going to be missing a single chromosome. So instead of having 46 chromosomes, that child is going to have 45 chromosomes in all its cells. And that's called monosomy. Mono means 1. Monosomy, we're missing a single chromosome. And that's going to cause some big problems. An example of that is going to be Turner syndrome, where we're missing a single chromosome. Now, that is non-disjunction during meiosis 1. Now let's go ahead and talk about what if non-disjunction occurs during meiosis 2. Let's see what happens there. So I'm going to go ahead, I'm going to erase this information. Erase this. And we're going to go ahead and erase this. So this is going to be non-disjunction during meiosis 2. That's where our mistake is going to be. So let's go ahead and get started. We're going to start with a tetrad, and everything's going to go well. This tetrad is going to separate normally into 2 dyads. There they are. And now we're going to have a failure to separate. Now again, because this is a rare event, chances are that both of these cells are not going to have a non-disjunction event take place. It could happen. It could happen. But more likely, only one of these cells is going to have a non-disjunction event. So let's go ahead and see what would happen if this cell had a non-disjunction event and this cell proceeded normally. So if non-disjunction occurred here, failure to separate, these two chromatids would not separate properly, and they'd all end up in one cell, just like that. This cell would be missing that chromatid that was supposed to end up here. And we're going to assume that this one proceeds normally, at least in this example. And this is what you're going to end up with. Now, here we have a normal set of chromosomes, N, plus one additional chromosome, so N plus 1. Here we have a normal set of chromosomes, but we're missing one, N minus 1. Here we have just a normal set of chromosomes. There's 1 plus 22 more, which I didn't show, but are in there. So we have a normal set of chromosomes. So N, and again, N. N plus 1, so we've got 23 plus 1 for a total of 24. N minus 1, so we've got 23 minus 1 for a total of 22. And then we have 23 and 23, what we should have. So in this case, this gamete, this sperm or egg, has an additional chromosome that it shouldn't have. If that meets up with a normal sperm or egg, we're going to have a child that has one additional chromosome. Again, that's going to be called trisomy, and that's going to result in big problems. Here, we're missing a single chromosome. So if this gamete meets up with a normal sperm or egg, it's going to result in a child who's missing a single chromosome. And that's going to be termed monosomy. And again, that's going to result in big problems. Here we have a normal gamete. It's got one of each type of chromosome for a total of 23. That's one complete set. And again, we have one complete set here. And we want a complete set because if a sperm with one complete set pairs up with an egg with one complete set, when those two cells fuse together, we're going to have two complete sets. And that's what we want. 23 plus 23 is going to make 46. And that's what a normal human has, 46 chromosomes. Now, can something like this happen? Can non-disjunction occur during mitosis? Now remember, mitosis occurs in somatic cells, cells that are not going to become sperm or egg. Those are called somatic cells, like nerve cells, muscle cells, skin cells, fat cells. Those are all somatic cells. What if non-disjunction occurs in a somatic cell? Well, depending on when it occurs, we're going to assume that it occurs later on in an adult individual. What happens? Well, you already have 100 trillion normal cells. So if you have a cell divide and non-disjunction occurs, then you're going to end up with a cell that is missing a chromosome and another cell that has an additional chromosome. Chances are those two cells are going to end up dying eventually. But that's not such a big deal because that's only 2 cells out of 100 trillion. Not such a big deal. And then those 2 cells will just be replaced by a healthy cell that goes through normal mitotic division. That's going to be a lot different than in meiosis. Because in meiosis, if one of these defective gametes pairs up with another gamete that's normal, it's going to result in a zygote, the first initial cell having too many or too few chromosomes. And remember, from that one initial cell that's imperfect, every single cell in that child's body is going to be a genetic duplicate of that one imperfect cell. So you're going to end up with 100 trillion imperfect cells on average. And that is going to be causing big, big problems. So that's going to be it for non-disjunction.